Good evening, everyone, and thank you for joining us today. My name is Urush Karatais, and I'm pleased to be welcoming you to this webinar on adopting sustainable colony management practices to enhance bee health. Biljana, your microphone is still on. Thank you. So each year, beekeepers across Europe and beyond face significant losses during the winter months, with causes ranging from varroa mite infestations and poor queen health to inadequate nutrition and increasingly unpredictable weather patterns. We asked members to report on winter colony losses from this winter, and we've received a few data. So, for example, in North Macedonia, they reported from 10 to even 90% of losses. From Ukraine, 20 to 25% of losses. Hungary, 30% of losses. Ireland, also about 25% of losses. And in Serbia, up to 50 or more. So as you can see, these numbers are quite high. And beekeepers each year have a bigger challenge to keep most of their colonies alive and healthy. So today we'll hear from experts who will share some practical insights, proven strategies, and emerging research aimed at improving colony survival rates through better preparation, monitoring, and adaptive practices. Whether you're managing a few hives or coordinating larger scale beekeeping, I hope today's session will offer some valuable tools to help protect your bees through the winter and support their resilience in the seasons ahead. The webinar is organized by the Scientific Committee on Bee Health, and the presenters are members of the committee. Professor Dr. Asli Oskirim, she's the president of the committee, Dr. Giovanni Formato, and Professor Dr. Ivana Tlag-Geiger. Before we begin with the presentations, I leave the microphone to the president of the scientific committee, Dr. Asli Oskirim, for a few introductory words. Asli, please.
Thank you very much, Urushka. Hello, everybody. Have a nice evening. And as far as I see, I know some names very well as beekeepers, and some scientists are with us this evening. So I am very happy to see your name, and I'm very happy to meet with you. Thank you very much for the European Beekeeping Association. So I'm very happy to meet with you. And as the president of Scientific Committee on Bee Health, we aim to help you suggest something on scientific base to help your bees and also your beekeeping management. We cannot create a miracle and to put our our abilities on your bees, but we can suggest something to help your bees, and so we can maybe make some comments about your winter losses or honeybee losses, because I'm sure that you are very sad about the colony losses. As Turkish beekeepers in Turkey, we are at the same situation. We had over the 50% of colony losses. So this year is not very fruitful for us. And all over the world, I think this year beekeepers will have some problems. So we are ready to share our knowledge and to help you with our scientific data to lead your management. But please don't forget that your management special for your technique or your apiary, your bee. So we couldn't have any idea about your apiary facilities or bees life cycle, and so that we will talk in general rule, and we must to do with in our apiary. So thank you very much for the organization again, and thank you, Ushka, and our president, Kostyan. And I would like to leave the microphone to the first lecturer. He is the vice president of the scientific committee on bee health, Dr. Giovanni Tomato. Thank you very much.
Hello, everybody. So nice to be here. I'm very happy to be part of the European Beekeeper Association Scientific Committee for Bee Health. First of all, can you hear me? Can you?
We can hear you well, Giovanni.
Okay, very good. Thank you. And now, please let me know if you are able to see the presentation.
We can see the slides in the PowerPoint, not the whole screen presentation, but maybe yes, we can see it now. Great, thank you.
Okay, great. So it takes some time to react anyhow. So I will present you the BDNet platform that is the first European platform for apiculture. And so, and how this platform can support European beekeepers in facing the mortality of the colonies. First of all, the BDNet is a network of beekeepers and advisors and all those people that have interest in the beekeeping sector. And the idea is to sharing knowledge on beekeeping concerning the best beekeeping practices. This platform has a repository platform where it can be possible for beekeepers to download the documents and see videos and pictures about diseases and practices. Okay, so as you can see here, we have a map with the partners that are participating to the BDNet. And so we have 13 national centers where beekeepers can work together and share their knowledge about best beekeeping practices. The project includes 18 partners from 14 countries, different countries. At the base of this platform, there is a multi-actor approach. So what this means? This means that beekeepers share their knowledge with scientists, advisors, technicians, veterinarians, and other people that are involved in the beekeeping sector, and adopting a bottom-up approach in identifying the most relevant practices to develop. They start identifying the practices that are more relevant through a survey. And the survey is specifically addressed to beekeepers. Once that the practices, most relevant practices are identified, those practices are written and shared among beekeepers, advisors, and other people that is involved in the beekeeping sector, and they are developed and placed into the repository. So it is important to focus the concept that the beekeeping practices they can be adapted to the different context that beekeeping has in Europe. This means that in the European Union we may have different bee genetics, we may have different beekeeping equipment and diseases, and so in each country the best beekeeping practices can be typical of that geographical area. I try to be more just to give you some examples. You can see different European countries, and so you may have different climatic conditions, different equipments, and so it could be that you can use some medicines, you cannot use other medicines because of the temperatures, especially for the organic compounds for varroa treatment, but you may have even different beehives, or you may have different bee genetics depending by the different subspecies of Apis mellifera that you have in your country. Moreover, even diseases are different. Are differently spread in Europe, and so you may have some priorities in some countries that are completely different from other countries. So this is the concept. It is at the base of the concept that the practices have to be tailored by the advisors at level of each country. So how can best beekeeping practices support beekeepers to contrast hive colony mortality? So in the left part of this slide, you can see the challenges for bee health and the issues that are responsible for mortality in Europe, the main issues. So we have the varroa mite, the pesticides, the wrong use of pesticides, the bad practices, and the climate change. So these are the main sources of mortality in our bees in Europe. So how can best beekeeping practices support beekeepers? To provide mainly knowledge and practice. So beekeepers can be supported by written practices or by videos, and they can improve their knowledge on how to contrast the above mentioned challenges for the bees. And just to provide you some examples, the best beekeeping practices include the strategies for a proper varroa management, as well as they include how to properly feed or watering the bees, or how to properly identify, place the apiary, avoiding pollutants like pesticides, etc. Moreover, in this way, beekeepers can control and prevent the mortality. To provide you some examples, verifying the levels of varroa infestation in their apiaries, adopting the proper strategies to control varroa, using the biotechnical methodologies in association with the treatments, and so on. Here are some examples of some practices that you can find in the BDNet platform. This is the explanation and different steps to follow for monitoring the varroa infestation using the sugar shake technique. You can see the different steps, and after you can see even the final values of percentage that affect your bees, and you can even understand if the infection rate is low, medium, or high, and an extra treatment is needed. Another example is how to adopt a similar technique that provide you the similar information about the varroa infestation level, but with the alcohol washing. In this case, as you know, this is a disruptive method, and so it is not possible to place the bees that are used for understanding the infestation level back to the colonies. And again, other steps up to the end. Another information you can find about best beekeeping practice on how to treat your colonies with oxalic acid, and how to adopt the biotechniques to improve the efficacy of your treatments. So here you have the example of the drone brood removal in association with the treatment, as well as the queen caging strategy to increase the efficacy of the varroacide treatments. In this case, it was the oxalic acid liquid sprayed on the bees after queen caging. So, where beekeepers can find the useful information about the best beekeeping practices, beekeepers can go in the website. They can register, and they can participate to the discussion on the best beekeeping practices, or they can go on the repository platform, where they can find the files of interest and videos and information that they need. So here you can find both the website addresses, as well as the QR code that you can use to download those information. Sorry, probably there is a microphone too close, I don't know. Thank you. So, about resources that could be downloaded by beekeepers, to provide you some other examples, there is a booklet on proper beehive management. It is in English, and we have a booklet for each of the year of the BeeTheNet project. So far, we have two booklet on the proper beehive management, as we are on the third year of the project, and it still has not been developed, the third booklet. And again, you can find useful infographics on. Sorry, is it possible to mute the microphones? Yeah,
Biljana, is it possible for you to shut down the microphones from other participants?
Wonderful. Thank you very much,
Ulska. Giovanni, sorry.
And no problem. And other information you can find on the planners, as an example, infographics on good beekeeping practices that are good practice to be adopted at the apiary level. As example, how to properly position the apiary, how to set up the apiary, as well as strategies to prevent and control honeybee diseases, like different kind of biotechniques, how to use organic antiviral treatments, and so on. Finally, please allow me to give you some very few slides about the survey. The survey is the tool that we use with a bottom-up approach to ask beekeepers which practices are more relevant for them, and it is very useful for us, as we can start developing those practices. So we are now we are now processing the survey of the third year of the project that is about the records that beekeepers should keep at the apiary level, the bee feeding, properly bee feeding and watering, and information concerning European foul brood. And I kindly invite you to participate to this survey. I will write in the chat these links where you can download the survey in your language, or at least in the language that you can be familiar to provide us information on the priority for European beekeepers concerning the most relevant practices to be developed in Europe. And I thank you for your attention. Thank you very much.
Thank you, Mayor. Thank you.
Now I put in the chat the link for the survey. Thank you.
Thank you very much, Giovanni. There's already a question for you, but maybe we leave that to the end of the webinar where we will have a Q&A session, if that's okay.
Wonderful.
And now,
thank you very much.
Yes. If anyone has any questions for any of the presenters, you can write them in the chat, or you can wait until the end of the webinar and ask with your microphone on. Until then, I kindly ask all participants to mute their microphones. And now we can continue. The next presenter is Dr. Ivana Tlaga-Geiger. Ivana, will you be able to... Are you here? I think she's here.
Hello, I'm here.
Hello,
Do you hear me?
hi, I can't see you. Sorry, that's why I asked.
Okay, so I will try to share presentation.
Ivana will talk about how to properly treat bee colonies, and I leave the word to you now, Ivana. Thank you.
So please tell me if you see the presentation is working.
We see the PowerPoint. Maybe if you turn on the presenting mode, we'll see it. Sometimes it takes a bit of time.
Okay, do you see the presentation now? No?
I don't see it.
Okay, it says that it is loading in the live mode, so probably it will last for a few minutes, or seconds. Nothing yet?
Not yet. Maybe a different window needs to be shared, Ivana?
I don't know. I'm in the right presentation. And I go to share. Here. So I will try one more time. Oh.
Okay.
Ivana, if it doesn't work, I can share your presentation and you tell me when.
Yes, I just want to ask if possible, Urshka or someone if you can, because I don't know why. Maybe it is overloaded with all the data. I don't know.
I wish I could. For some reason, I can't. My button for sharing isn't working. So Giovanni, maybe if you
Okay.
have.
So maybe I have the same problem. I don't know.
I try. Okay. So screen. Okay. Can
Thanks,
you
Giovanni.
can you see it? Okay.
Okay.
Presentation mode. Okay.
Please
go ahead, Ivana.
Thank you.
Please.
Thank you.
Thank you. Welcome.
Okay. So firstly, I would like to greet all of you, this time from Croatia, from Križevci, and I'm very grateful to the European Beekeeping Association for the opportunity to talk about how we can properly use the medicines if we want to treat the bees. So maybe next slide. Okay, thank you. So if we talk about the strength of the honeybee colonies or their immunity, so we have to say that there is many different influences which depends about the health of the honeybee colony. So firstly, we have to say something about the honeybee colony status. So that's is linked with the strength of the colony, then the immunity status of this colony, which is usually linked with the microbiome in the intestinums. And also it is very important to say something that we have always some kind of infestation with the varroa mites. So that's all is influenced, of course, by the environmental impacts. And here we can say that the let's say the most important environmental impact is the availability of the natural food, and not just the quantity, but also the quality and diversity of that food, which is primarily depend about the weather conditions, and of course different pesticides which are present on the treated crops, field crops. Also, there is the human influence, and when we speak about the human influence, that we have to say that it really depends about the applied beekeeping practices, biosafety measures, about the knowledge, and maybe some notifiable or notes about the properly using a different kind of medicines, food additives, and other substances, which sometimes can be inappropriate. So you see that all these influences are overlapping. So it is a really let's say industry under the sky. So bee management or the influence of human is sometimes very, very important as well. Okay. So in the last decade, especially, we are often heard about the honeybee health crisis. So, and the main of the reasons for that situations are noted as the climate change, then the problems with the nutrition, diseases, of course, the influence of pesticides and acaricides, and bad beekeeping practices. According to these pesticides and acaricides, I have to say that, let's say the hive as a part of the equipment in beekeeping don't have a metabolism. That means that everything which the bees take accidentally during the flight activity in the hive, or all the substances which we are using, especially against varrosis, will stay in the hive. So where in the hive, as you can see, the bees are living together. So the adult bees, their brood, their food, and it is all situated in the nest, which is built from the wax. And wax is a substance which really likely want to take all these substances and contain it for a longer time, maybe a few years. It can have some effects on the bees, on the quality of the bees food, but also for the food for the humans, as you know that the bees are animals which are producing the food for humans as well. Okay. So when we speak about the honeybee diseases, parasites and pests, we can divide them in a few groups. So in the first group, we can say that we have the notifiable disease, a very serious infection disease like the American foul brood, then the European foul brood, and also some of the parasites and pest diseases as the varrosis, then small hive beetle, or tropilelaps. So for that diseases, there are some special eradication measures and sanitation measures, which are usually prescribed in the international and national levels. So we cannot use for them in the European Union any kinds of authorized medicines. Then we have the group of the diseases. We call them usually the conditional diseases or secondary diseases. So they are also can show some special symptoms, but especially after the decreasing the immunity system of the bees. So the causative agents are present in the apiary, in the hives, but they are not showing the clinical symptoms before they really have crashed the immunity system. For that diseases, we also don't have the authorized medicines, but we can use some of the technical measures at the apiary and help the bees to get them strength and the immunity system enough for recovering from the diseases. And I have to say that there is also some group of invasive species and the parasites where we have to implement different traps and make some measures to just to keep their number in some level which will not produce much damages at the apiaries. The next one. Thank you. So at the end, if we just put the line, we can say that the only disease which is caused by the obligatory parasitic mite, Varroa destructor, it is called varrosis. Of course, in the situation when we have a very high infestation levels with mites. So in that case, we have to use or are obliged to use some kind of veterinary medicines, which will allow in the combination, of course, with the biotechnological measures to hold this threshold or number or state of the invasion with varroa mites on the proper level, which will allow the usual and productive life of the bees, but we are not able to eradicate these mites. So you already know that varroa females live as parasites. They are fat body feeding parasites, and they are parasiting on the all stages except the egg, on the undevelopment immature stages, but also in the adult stages of the honeybees. So because this is the kind of the feeding which they are performed are reason for the getting some mechanical damages on the bee's skin, and we call that skin the exoskeleton. It is pretty hard and it's not so elastic. So in the most of the cases, this wound called the feeding place or feeding wound will not be sticked together, and it will be used as an open door for different kind of pathogens during the life of the adult bees. So because of that, we have, let's say, the crash of the primary individual immunity level or barrier on the individual bees, and it is also used as an immunodepressive factor for the whole the colony, not just the individual bees. So it is often called as a trigger for the appearance or the outbreaks of the different, especially these secondary diseases, like whole list of virus diseases and other conditional diseases, but sometimes it can also be a nice, let's say, vehicle or vector for the serious diseases, for example, such as the American foulbrood. So here on these photos, you just can see a different kinds or different symptoms or signs of virusological diseases. Okay. Thank you. So let's say that when we are talking about the controlling of varroa infestation, we have to be very careful during the whole year. So that means that we must monitor the varroa mite infestation rates during the whole year. So not just during the active season, not just during the summer or the early fall, but during the whole year in many or multiple times. How we can do that, especially in the apiary. So there is a few already mentioned methods by the Giovanni. So like the field sugar test, so where we can estimate the number of the varroa mites in some standard sample. So that is some 2050 to 250 or 300 bees. So which we will cover with the powder sugar, and it really depends about the term in the year or in the summer months where we are doing that. And for each of these months, we can estimate what's really stay in the hive. According to that number, of course, then we can decide if the treatment is needed immediately or we can delay it. And of course, we can decide about the possibility what kind of medicines or different methods we will use as this treatment. Then there is a possibility for diagnostic treatments with one shot, some of the acaricides, or maybe monitoring the natural mite fall during the 8 or 10 or 40 days and see where is that threshold to find a right time for the treating. So the next one, also there is a possibility, of course, for the laboratory methods, as we already see. So let's say that the first rule is that if we have the intensive flow of the nectar from the nature, then we are not able to use any kind of acaricides or chemical substance, because we want to avoid their harmful or hazardous residues in the food, and of course in the food for the bees primarily, but also for the humans. In that case, we can use apitechnical methods. So they are mostly based on the brood removal, then isolation of the queens of different kinds of trapping combs, like this drone built frame. After which the cells are closed, so we will just cut these parts of the brood and melted it. So in a mechanical way, we will remove some of the mites from the colony. So we can say that that is a whole group of different methods, which is based on the natural mite traps. The next one. Okay, so the first rule we already said, for the European Union, the most of the members of the European Union countries, the only acaricides which are permitted by the legislation and according to the national regulation of the veterinary medicines, and of course the cascade principle, which is also used in some countries. So the next situation is that the most of the authorized VMPs must be prescribed by the veterinarians, again, according to the national regulations. They must be applied outside of the nectar flow, and they must not be toxic to the honeybees. That is really depending about these outside weather conditions, temperatures, and so on. And they must be used according to the manufacturer's instructions if we want to achieve some optimal effects. So I will say a little bit about that later. And of course, that all the medicines must be used by the care to decrease the risk for the health of beekeepers. So for sometimes they have to really use protective clothes and everything. And of course, all these used acaricides must be effective on the phoretic phase of the mites. So that are the mites on the adult bees, because most of the active ingredients doesn't work or do not work in the cells, in the covered cells. So only it is known that the formic acid is active on the mites within the comb cells. Of course, there is some rule or practice that you have to change the active ingredients of the medicines from year to year to avoid the antimicrobial resistance. And different active ingredients, as you will see, are used in the different carriers, which are with the aim of sustained release of different treatments. Okay, the next one. So if we want to use some kind of medicine, we must know some of the facts. Firstly, when is the right time for using it? So I already said, if we monitor the infestation level of varroa mites, and of course, we know that we are in the part of the year where we don't will use it for the nectar or honey flow. Then we have to choose what kind of medicine. So that's really, it's very hard to tell any of the active ingredients or any of the name of the products, because the medical history that you are applied and your apiaries, the best knowledge is from you. So what kind of medicines did you use till now? What was the efficiently? How you really estimate that efficiency of the mites? Did you checked the fallen mites status and so on? How to apply the medicine. So it is very important to know in the which place in the hive you will use some kind of medicine. That depends about the active ingredient, but also the carrier which is used to take these medicines in the hive. So sometimes the same active ingredients, the recommendation is to use it in the different parts of the hive. So sometimes is it, for example, in the middle of the frames with the brood, and then other times for same active ingredient is strongly outside of the area of the brood. So we must be very careful and read the instructions for using. And also we need to be able to properly interpret what that instructions really means. So the next step is the dose of the treatment. Dose is very, very important. And of course, in the beekeeping, how long does this treatment last? So most of the authorized medicines in beekeeping are aimed to be used for so-called chronic treatment. So some period, and the whole time the carrier must ensure that you will have the small amounts of active ingredient which will be realized during that treatment lasts. So I have to say that when you are thinking about treating or you are treating, you must be in connection with your neighbors. So it is not a situation that in beekeeping we are working individually, because the strength of reinfestations is pretty high. So if we want to do a really good thing, not just for our apiary, but also to the neighboring apiary, it will be very good if we can recommend the model of simultaneous treatments for the honeybee colonies and apiaries in some area. So that will give the best results. It is not so important what have you choose as a medicine, as a different beekeepers, but if you do it simultaneously in some, for example, 2 weeks period, then the effects will be for sure better. Okay, the next one. So here is one video. I hope it will say everything. Okay. Do you hear anything? No, I don't know.
I can't, unfortunately.
Okay, so here I want to show you what is really important during these treatments. So, of course, that you will choose some of the carriers. So the most group of active ingredients which are authorized, we can divide them in three main groups, let's say like that. So the first one are the hard acaricides, which are based on a different synthetic acaricides. So that is the amitraz, cumafos, flumetrine, tauflavolinate, and all of them coming on the carrier of the strips. The strips of so-called smart plastic, which are able to work during the chronic period of almost 42 to 45 days. Then we have a pretty big group of the active ingredients based on the thymol. And there is also a few kinds of carriers. So we have the thymol in gel, in tablets, in the cellulose sponge, and we must very, very careful because of the outside temperatures during application of these thymol preparations, of course. And the last group is a pretty big group of the active ingredients based on the organic acids. So it is very often used the oxalic acids, then these lactic acids, and they are also part as an active ingredients of the authorized products. Sometimes the beekeeper, no. I don't know what's happening. It's
no, I'm sorry. I'm sorry, Ivana. Probably it's too much work for my laptop. I'm sorry. It's not able to show the video.
Okay, so it's time for you as also as for me to find some new laptop in the near future. Okay, so for example, just for example, I will say that I often heard that it is resistance, this medicine doesn't work, and so on. And the first question is, did you check the efficiency? So in many cases, I don't have a positive answer. But what is the places, or where are the places of the risk for the development of the resistance? They are always somewhere in the application of the medicines. So hyperdoses or hypodoses, so much higher doses, or some doses which are not as recommended, is one example. So then if we decide to say, okay, I don't have a really strand colony, it is a pretty weak, I will not use, I don't know, 2 strips as is prescribed, I will put just one. Or maybe if the treatment lasts for 45 days, for example, and then the beekeeper says, okay, but I will left them the strips in the colony because there is a little bit of the substance, so they will have it for the next period. So that are the places for development of the resistance. There is a small substance, but not in enough concentrations that it can kill the mites. So what I want to emphasize at the end, that we have a really good or really big obligation as a public health. And again, the hive doesn't have the metabolism. So everything what we put in the hive will stay in the hive in some levels for a longer time, and can be a negative effects on the queen, on the brood, on the house bees, the young bees, not just for the adult bees or foragers, or I don't know, possibility for some residues in the hive. The next one. Okay, so what is really important, just like a conclusion, that we have a communication, especially with keeper, with the beekeepers, the other ones, but also between the all kinds of stakeholders which are included in the apiculture, and of course the educations, which is really important, like long living education patterns. Okay, the next one. Okay, so if we will try to think about our tasks at the apiary, hopefully that will not be just happy as for successful control of varroa mice on our apiaries, but the neighbors will also be happy. The next one. So it is the last one. I hope that I didn't go too long and too far, but my message is, let's protect the bees together, in, of course, always in the service of One Health. That means that we need to take care for the health of the animal, but also the environment and the humans. Thank you.
Thank you, Ivana.
And thank you, Giovanni,
Thank you very
for the
much,
presentation.
Ivana. And thanks,
Welcome.
Giovanni, for offering
You're
the
welcome.
presentation.
You're welcome.
Thank you, Ivana.
Thank you, Ivana. And now I give the word to Asli.
Yeah,
She will talk about the integrated varroa destructor management. We are already getting some questions. So again, if anyone wants to ask anything to the presenters, please write the question in the chat. You will also have a chance to ask after this final presentation from Asli.
Okay, thank you very much, Kuchka. Can you see the presentation in full screen? Can you see?
We see the PowerPoint. I don't see the full screen yet.
Yes. Okay. Again, I try it. Now? No?
Does anyone see it full screen? I don't.
No.
No?
Yeah,
Okay.
we just we see the screen.
Okay. Again, I share the window or all screen. Okay. And now maybe you can see my slide. It's okay now?
Yes, now it's full screen. Great.
Okay.
Thank you, Asli.
We managed it. Thank you very much. And thank you for the nice presentations of my colleagues as a bee scientist, because we are bee scientists, and bee science is also multidisciplinary area. So some of us are veterinarians, some of us are biologists and chemists and zootechnics. So we are a big family on Science Day to work on bees from different sides. That's why, as a biologist, this evening, I would like to take your attention to the bee biology. That's why I wrote a title as Integrated Varroa Fighting or Control or Management, not only the treatment, because I would like to emphasize that Varroa destructor is a parasitic mite. So it's a parasite. Means that we should we should recognize that what is the difference between parasite and pathogen, because treatment is made for pathogen, but the parasitic story is totally different from pathogens. That's why we need to apply integrated, sustainable varroa control, fighting, or management in our colony. This is the different philosophy of the varroa management. A parasite in an organism that lives on or inside another organism. So varroa mites lives on our bees and our bees larvae. And our bees called the host and gets its food and at the host extent. Parasites can harm their host, but they don't want to kill them, because if they kill them, means that they can lose their host, so they cannot be survived, because they don't have a free life without their host. So this is the critical point for the varroa control of varroa management in our hive. Please keep it in your mind. Varroa cannot be survived without bees, because they are parasites of bees. They rely on the host to survive, reproduce, and sometimes even move around, like on our foragers. So they can fly with, by means of our foragers. So if there is a good balance between host and parasite, or there is environmental factors are suitable, not too hard, not too changing, like climate change, or there's no environmental challenges, and there is no other pathogen or immune weakness, parasites don't kill their host unless there is a good balance and there is no extreme environmental factors, and there is no other pathogens or immune weakness of bees. So please consider that the challenge of the bee on the bee, from the environment inside the hive, and from the beekeepers with their colony management, from the nutritional materials, and also the bee genetics, because bee genetics is also a very important challenge factor on bees, because the adaptation of the genetic factors or ecotypes of honeybees and the environment are very important point to be alive, to be survived for bees. So if I can explain that the parasitism and the parasitic life rules to you, I can show you that the the differences between the parasite and pathogen. Parasitism or parasitic life is the long-term biological cycle on the whole, but the pathogen can be survived in a short-term activity on their organism. When the pathogen enters one organism, the pathogen can shoot very suddenly, and the acute, very rapidly, acute effect on the organism. It doesn't matter if it's human or honeybee, in any kind of organism. Parasitic life is a passive life. A parasite cannot be determined, doesn't want it to be determined by the immune system. That's why they cover them. Sometimes they cover into the cysts, or they use the cell surface on their surface to escape the immune system cell. So they would be a passive, but pathogens are always aggressive. and they are invasive, and they want to, invade all tissues or all other sides of organism. And in parasitism, parasites are host-dependent obligate life. Okay, some parasites are not obligate, but some of them are obligate, that varroa is included. Yeah. But the pathogens can be survived in a free. So they can be survived outside the organism, and then they can synthesize their proteins and other substances easily. They don't be survived as a host dependent. So if varroa destructor is a parasite, means that we have to fight with varroa in long-term periods, not only the winter time or springtime, as mentioned by Ivana and also Giovanni. And our fighting methods should be in accordance with environmental conditions and also in compatibility with bee biology. We should know the bee biology and biological life cycle, because all parasites follow their host biological life cycles, like varroa. So as you see, varroa strictly follow the bee biology. So when the queen bee laying egg, all female varroa go enter the brood cells, and they suck on the larvae, and with larvae they get out to the cells and move on the adult bees. So they follow the bee biology. And every year, or for decades, varroa always living in our hive. But some years, our colony losses rate are very high. So we always say that, okay, we lost our colonies because of varroa, but we are used to live with varroa for many years. So why some years we have very high rate of colony losses, some years not? Why? Because the impact factor of varroa affected by the other factors, as I mentioned before, other challenges, environmental, nutrition, and the human touch, and the beekeeper's touch, and other pesticides. That's why some years, parasite is a parasite, it's stable, but other factors can move forward to parasite effect. This is the result. So some beekeepers think that, okay, scientists always say that the cause of the losses is varroa, but something would be different in this year. No, there is no difference. Some years, because of the climatic conditions or other challenges, has more effective than the previous year. So with this knowledge, what is the integrated varroa fighting or varroa control, IVC or IVF? Fighting is my term, telling the truth, and I like it very much. We should keep the varroa fighting in long-term period in every season. For this aim, for instance, in winter time, we suggest you, regarding the literature and all scientific research, to use organic acids and formic, oxalic, lactic, and acetic acids. And so far, all the papers showed us that there is no resistance to oxalic to organic acid, because they are operating, and the ingredients do not leave. Sorry, microphone. Please mute it. ingredient substances cannot live very long time in hive. So organic acid can be recommended, suggested for winter time. But please consider humidity, because we use organic acid in sorry.
Excuse me, participants, can you please mute your microphones?
Thank you very much. When you are using organic acid, please consider, please observe the humidity level of the weather, environment, and also in the hive, because the dropping or evaporation or sublimation methods increase the humidity in the hive, and the humidity means that you can open your hive to the fungi infection like chalk brood or stone brood, and also other microbial infection, because humidity can transfer all things to the bee, the extreme conditions, and bees can be affected, all environmental challenges under the high humidity condition. And in springtime, you can create the drone larvae cells under the I don't remember the equipment, syrup box, or you can leave the frame, empty frame in the hive, so bees can create the drone larvae, and you can harvest the drone larvae, and you can use the drone larva cell as a trap for varroa mite. So in this piping method, your target group is larvae. So you can clean the varroa mites in the larval stage inside the brood cell. And the other method is queen caging or brood interruption and the brood holiday method. In this method, the target group is also larvae, because if you cage your queen or if you take out the brood, you can catch all varroa mites inside the brood cell. So you can kill or remove the varroa mites from the larvae as well. And the third method is in springtime. We are in spring now. Powdered sugar method, but be careful to use the powdered sugar in the package. We don't recommend to beekeepers that the ready-to-use powdered sugar, because sometimes they can contain the starch in matrix. So it means that it is there is a risk to include your honey, because after spring, summer will come and nectar flow will come. So the starch can destroy your honey with their residue. In this method, the target group is adult bees, because when you use the powdered sugar on your bees, you encourage your bees for grooming, and they clean each other by grooming, so they can clean their varroa mite from each other. So it's very useful method, because the springtime is also feeding time. So if you use exact powdered sugar and the sucrose, when you crush the sugar crystals to make powdered sugar, it would be very useful in springtime. And the fourth method is pollen trap, using pollen trap. In this method, target group, your target group is adults, but adult bees, but the foragers. Because as Ivana said, that please follow your neighbor apiaries, and simultaneously varroa treatment would be useful. Yes, it's right. So if you use some varroa treatment methods, just after that, your bees can fly to flowers and contact with other bees, your neighbor apiaries, so they can be infested again on the flower or on the watering box. So they can take extra varroa mites into the hive. So if you use pollen trap, not the pollen flow time, in different periods, because bees always need pollen. So it's a critical method for you. You should be experienced to use this. And they just come when they enter, while they entering the hive, you can groom your foragers and escape and move out the varroa mites as well. And the fifth long-term fighting method would be long-term release and temperature-independent organic acids. These organic acids effective mechanism by contact, not the evaporation. And the target group is adult bees. So some companies add the oxalic acid or combine the oxalic acid with glycerin or chitosan. Chitosan is a monomer of the cuticular base of the honey, the cuticular surface of the honey, so sorry, not base. So chitosan or glycerin make the oxalic acid in a gel form. So oxalic acid cannot evaporate very easily in the gel form, because chitosan can keep the oxalic acid inside it with the polymerization. So in the gel form, it's a long-term release, the effect of oxalic acid by contact. So this is the another this would be another method for varroa. In summertime, there is a nectar flow. So you cannot use pesticides and other feeding methods, because there is a residual risk in honey. So sometimes beekeepers can use some organic substances by spraying, for instance, thymol or chitosan as well on the frame. By this way, they can move, they can get out the varroa mites in summertime. But it's not suitable to use other veterinarian substances during the nectar flow, because it's very dangerous or risky. And I consider autumn season very much because it's very important and critical season for beekeeping in autumn time. Because if you prepare your colonies very well in the autumn period, means that your colony can be very beautiful, not only for winter, but also for the next year. That's why the autumn period is very important, and also the critical period, because in autumn season, you need young bees, and colonies need pollen for new generation, and need strong immunity, so they need pollen very much, and need long lifespan, because you produce the new generation, and they should be survived during winter, and after winter, till the first generation of your colony, they should be on the brood cells. So they need long lifespan. That's why you need young bees without varroa damage. This is the very important and critical point. Please imagine the varroa bite like in the slide, the knight behind the man. So all varroa bites damage on your young bees in the autumn time. So there is a very big wounds or holes, and they open the secondary infection. And during the winter time, because of the low temperatures and immune weakness and other challenges, the wounds can be open to secondary infections. So just after the winter, you can come across with the high mortality or high rate of colony losses. That's why in autumn time, please follow the weather conditions and population balance. I mean that please observe that if you have young bees without the mate and varroa infestation level is low, or you still have the old bees from the summertime, even though they will die. So the target group must be adults, including foragers and also the young generation. And you should concentrate on to cover their wounds and varroa damage. And also, in contrast of the springtime, you should avoid from using pollen trap, because in autumn time, your colony needs more pollen than the springtime, because after spring, summer will come and they can support their body. I mean, honeybees can support their body by nectar. But after autumn, winter will come, so honeybees will need immune response. So they will need protein, and the pollen contains protein, not nectar. So please avoid from using pollen trap in autumn time. And also just after the harvest of honey, you can use the veterinary product, not the resistance. I support simultaneously veterinary treatment in the colonies, in the big areas. I totally agree with Ivana, but I'm not sure that the selected active ingredient, for instance, in the region, okay, beekeepers or association or government decide that amitraz will be used for the simultaneously treatment, but some of the bee colonies would be resistant to amitraz. Some of the bee, not colonies, varroa mites would be resistant to amitraz. So if we decide to use only one ingredient in 1 year, some of the colonies cannot be affected from this veterinarian substance. So it should be well organized, but I totally support the simultaneously treatment as well. So regarding the integrated varroa fighting, I kindly remind you that please keep your mind, varroa destructor is a parasite. So there is no way to use only one substance against varroa, or only the limited time for varroa fighting. You have to use all techniques together, but in compatibility with the bee biology. So you have to follow the bee biology as well. And thank you for your attention. Hopefully, it would be useful for you.
Great.
Okay.
Thank you very much, Asli. Thank you to all of the presenters. I think we've heard a lot from different points of view on how to best keep our bees. So if you all agree, we can maybe go to questions. Does anyone want to ask a question? You can turn on the microphone, maybe raise your hand first. You have a button on the top to raise your hand. And while we're waiting, I can read the question from the chat. It's for Giovanni. Someone is asking how the situation is with the small beehive beetle in southern Italy after it reappeared last year. And the second question from the same person is if countries that are outside the EU, such as Albania, can be included in your projects.
Yes, of course. Thank you very much, Uska, and thank you for the participant for the questions. So the first question about Etina tumida, the parasite is spreading in 2 regions now. The one is Calabria and the other one is Sicily. The difference respect to the past is that according to the new animal health law, it is not mandatory to destroy the apiaries in which Etina tumida appears in new areas, and so it is slowly spreading. This is the final result, even though it is fortunately not so fast. Of course, it depends a lot by the movements that in the area in which Etina tumida is present are forbidden. But of course, it depends even by beekeepers that they do not move colonies. It has been told and explained to them, but of course, it is even their responsibility not to move colonies from one infested area to the other, and not infested. And another thing about Etina tumida is that fortunately it seems that it is not so dangerous as in other countries. It is similar. The damage that it causes is similar to the wax moth, more or less. But of course, some effort have to be taken, keeping the colony strong and controlling the parasite during the inspections that are done in the colonies. I hope it is enough for the question about Etina tumida. And concerning the other question from Albania, yes, of course, we are very happy to have you on board and to have your documentation about best beekeeping practices to be shared with other beekeepers on our platform. It will be a pleasure. Moreover, you can contribute to develop the manual for advisors and that are people that teach beekeepers, and in the manual it is kept into consideration the best practice adopted in different countries. So we are very interested to know what you are doing in Albania to keep the bee healthy and to control the bee diseases, and to see if in your country you make the same as in other countries, or you may even have some ideas to better improve our strategies, or vice versa, of course. So please, I write you my email and send me an email and we keep in touch, okay?
Great, thank you, Giovanni. Someone was asking also about Bulgarian language for the survey, but it's not available in Bulgarian, right? The survey.
Unfortunately not, but it is in English. If you can translate in Bulgarian, it can be spread even to Bulgarian speakers. But we don't have partners from Bulgaria. So if you can take care of translating, If you want, we can provide you a draft done by the simultaneous translation of Google Translator, and after you revise, verifying the terminology is correct. Anyhow, you have my email, and write to me, and we can verify what to do. Thank you.
Great, thank you, Giovanni. So if anyone's here from the Bulgarian Association or from any association from Bulgaria, if you're willing to help with translation to make this survey available to your beekeepers in your language, contact Giovanni, please. There will be contacts in the chat, right, Giovanni?
Yes, sorry, probably I was starting. No, yes, I wrote it, yes.
Okay, great. So you
Yes.
can find it in the chat. And now we have someone who raised their hand. Feel free to unmute and you can ask your question, please.
Hello, everyone. Can you hear me?
We can hear you.
I'm Matthias Manch from Germany. Thank you very much for your presentations. It was very interesting from all of you. I have one specific question to Professor Dr. Asley. I found the part about using pollen traps to fight Varroa manifestation very interesting. Could you briefly explain how that works, please?
Yes, especially except from the pollen flow time, because at the early spring, okay, we can use pollen trap for collecting pollen. And at the bottom of the hive, you can see some varroa mites as well, because the pollen, they can come to the hive. But after that, in the nectar flow time, we use also pollen trap as well. We open them to um, to groom the foragers when they enter the hive. They have to move among the hole, the pollen trap hole, so they can help the varroa hold down on the bottom side of the hive. So there is some studies belongs to me and also some other authors to compare with them. The rate varroa mite fall is not so much rate, but it helps for forager contamination.
So at what time do you use that trap for the varroa mites? Is it just during the pollen collection phase, or is it a specific time when you have to use it?
No, except from the pollen collection, because the pollen collection period, we focus on the pollen collection. But after that, we don't need to collect pollen anymore. And especially when the nectar flow time, the summertime, you know, we don't concentrate on the new generations, because nectar flow and our foragers works more frequently, fly more frequently to the flowers. That time we use pollen trap to clean the foragers when they are entering the hive. Summertime, the nectar flow time, not the spring. Okay.
Thank you.
You're welcome.
Thank you. Then we had questions in the chat and Q&A section about the resistance. I think all three presenters might be able to say something about this. So the first question was if there are any known varroa resistance on formic and oxalic acid, if there are any studies which confirm this and that they're available, if the studies are available.
So far, I couldn't, I didn't read any article about resistance to oxalic or formic acid, but regarding their chemical molecular pathologies, they can be easily evaporating and also destructive. Not destructive, it's not the right word. But anyway, when the substances can live in the hive in long term, means that there could be a resistance against the substance, because it means that the varroa can introduce the substance and react the substance. But if the substance comply, or the other substance keep the organic acid inside it, means that varroa cannot have an opportunity to meet with the substance for a very long time. Or that's why I think that at least for the near future, we don't have any resistance problem with organic acids regarding with their molecular facility. Giovanni, would you like to say something about it? Or Ivana?
Even by my side, there is no knowledge about resistance of varroa against formic acid or oxalic acid.
Yeah. We didn't read anything.
Fortunately.
Fortunately. Yes.
Thank you. And Ivana, then there was a question specifically for you, whether you think the frequent use of synthetic acaricides for many years in a row has reduced their effectiveness in the fight against varroa, and perhaps that varroa has developed resistance to some types of preparations, so medicines based on substances such as amitraz, flumethrin, coumaphos.
Sorry, what is concrete question? Is it in the chat? No?
It's in the Q&A section. And it's if you think that the use of synthetic acaricides for many years in a row can reduce their effectiveness, and that the varroa builds up resistance to active substances such as the mentioned.
For all known synthetic acaricides was proven the resistance in some geographic areas that is known. And because of that, it is very important to do the proper treatments, and not just the rotation of active ingredients, but because in the literature there is some data that it is possible to use the same medicine based on sub-active ingredients if it is used properly for a few years in a row. But most of the authorized products are produced in a way to be safe for honeybee products. So mainly the beekeepers are not very satisfied if they don't see that all varroas fall down in a day or a few days. So that is the problem, that some of them decide to use something else. So they left this authorized product in the hive and put something else. And the most of the, let's say, homemade acaricides are also the mixtures of these active ingredients which are present in the authorized medicines. So that is also the problem with the long-term residuous development in the different areas. And also this, we can say the cocktails of acaricides, which are also pesticides, in the combination with the pesticides from the field, can have a really negative effects. And if you heard these big colony losses in the United States for the last year, so they really don't know the one reason, but these cocktails of pesticides and insecticides are one of the mainly written now in these days in the different comments and forums and so on, and also as a result of laboratory examinations. So we have to be very careful So if we just on our own responsibility take a higher doses, mix the products. So I'm just scared with which ingredients, which medicines we will treat our bees for a few years, for 5 years or 10 years. So maybe anything else will not work. And that is the reason and the base of my speech. I want to just take the line that it is time to think what we are using and how we are using the medicines in the beehives.
And also
yes, that is very important. Thank you, Ivana.
Their synergetic and antagonistic effects together. Some substances, some ingredients can be very useful and no toxic effects, but when they combine in the hive, when they meet in the hive, they can work together. So that's why it's time to think about it.
Okay, thank you. And then we have maybe just a quick answer to, because it's connected to everything that we've been saying just in the last few minutes. Someone is asking, if they treat with oxalic acid, do they need to change the treatment with something else every 3 years?
So I will just say a short comment. So maybe maybe it's good to change the way of application, if not the active ingredient. If you are satisfied with it, it is organic acid. So the possibility for developing the resistance is lower than the synthetic acaricides. But there are different authorized products based on organic acids as well. So maybe sometimes just to interrupt routine and change the carrier, for example, or weight of the application.
I would like to make a short comment, and I would like to make a simple calculation. If you have 100 varroa mites in your hive, when you use integrated varroa management in your colony, for instance, the powdered sugar method can reduce 10 or 20% of them, and pollen trap can reduce 10% of them, and drone brood cell methods can reduce 20% of them. You can have 50 varroa mites in your hive. And if you continue with other apitechnical methods like queen caging, heat shock, or brood interruption, you can reduce the infestation level during the whole year. So you don't need so much synthetic acaricides or other substances. They can be supported by other methods. That's why we recommend you to use integrated several methods during the biological cycle of honeybee colony. Please don't trust only one ingredient or one substance to control the varroa infestation level in your appearance. You have to use different things. Depends on the season and depends on their your colony and also the environmental challenge. Thank you.
So in connection to what you just mentioned, Asli, there are also two questions regarding the reduced use of varroicides. So there is one on how do you see the possibility of improving biotechnical measures and breeding in order to reduce the use of treatments. And there was also a comment that how will bees develop resistance if we continue treating all the time, maybe. So this is, if we can say something very quickly on possibilities to reduce treatment.
That's why I emphasize that varroa is a parasite, and I don't use the treatment as a term, because treatment can be made by some substances, treatment, but the control or the fighting or the management, including the all different kinds of methods. If you use all the time, whole year, if you use different chemicals, yes, you can have the varroa resistance, because you may have resistance problem and also residue problem in your hive. That's why I recommend the beekeepers to use different techniques, some physical techniques, technical techniques, and also synthetic acaricides after harvesting the honey and the organic substance. In different periods, different time, and different methods together, not only the one thing.
Thank you. Would Ivana or Giovanni would like to add anything? There was also a comment on Colos. I know that our presenters are very active members.
Yeah.
And there someone pointed out that there is an organization called Colos that has an ongoing surveys for many countries on colony loss, so they have statistics for each year.
Yes.
And that there are national coordinators, and maybe Ashley, you can say more about this.
Yes, I am also the vice president of Colos organization now, and we are proud of our task force, the core project, not task force, the core project, monitoring. And for several years, more than 15 years, we monitoring the colony losses in different countries, and we have national representatives of each country. But some years we cannot make questionnaire, apply the questionnaire among the beekeepers, because beekeepers claim that they cannot take some results from the questionnaire, and they cannot prevent their colonies from the losses. So sometimes it would be difficult, and we try to different ways to collect data, but we are working sustainable in a sustainable way on the monitorization for colony losses. Thank you for your contribution, our Colos member. Thank you.
Great, thank you for these additional information, Asli. Maybe we have time just for two last short questions. One is about the heating cameras for what is it? So on larvae, so that you put the comb into the heating camera, if we know anything about effectiveness against varroa. And the second is about glycerin method with oxalic acid. If maybe any of the presenters can say just a few sentences about it.
Yes, of course. Concerning the glycerin application with the oxalic acid, it is a methodology that allow and that is low release of the oxalic acid. So it is very useful for organic treatments, and the and it can allow a longer contact of the oxalic acid with the varroa mite. It can be useful, especially in periods in which you have a brood and you need a longer treatment of the oxalic acid, as the trickling the oxalic acid it allow the acaricide efficacy just a few days after the treatment, but after it stops. Adding the glycerin in strips and soaking the strips with oxalic acid allow a slow release of the oxalic acid. So it is, in my opinion, it is very good that we are starting to have commercial strips that allow standardization of the treatments and allows even to study this kind of application in more standardized way. Anyhow, it's good to have other organic treatments available for beekeeping. I don't know if it is enough. So the
Yes,
glycerin.
thank you. There was one
Yes.
question, additional question on the pH change. So if a long-term disruption of the hive's pH level has any negative impacts on the colony?
Not so much on the, I mean, it can be an impact on the colony. It can be toxic for the weak colonies. It can be, and it can even be, it can even impact the acidity, the free acids that are in the honey. A little, it can impact. So this is why I say that to have standardized medicines can be useful to go more in deep on these aspects.
Yeah. And
Okay,
also,
thank you.
my team and myself is working on studying on, or was working on the phytocan and oxalic acid. And this is also the process on the licensing and standardization for a commercial product. So in the glycerin formula and also phytocan, they are the same effectiveness for pH degrees, and but the low pH also induces the lactic acid bacteria for growing, and it's useful. Not only the side effect, for other side pH it can be harmful in the long process, but for microbiological approach, it's not very bad for the colony. If the colony is strong, it's not a very bad thing. And also, phytocan is a prebiotic, so it can be picked by beneficial microorganism. So this is another formula, and both of them are the gel form, so they can prevent the side effect of the acid. They keep their evaporation temperature. That's why it's
Great, thank you. We are running out of time slowly for our webinar. In the name of the European Beekeeping Organization, I would like to thank to our presenters for a very comprehensive presentation of the topic, for answering, I think, most of the questions. Hopefully we will answer some more questions at our future webinars. We are planning to be in communication with you beekeepers more the next fall and next winter. So please follow our webpage, follow our Facebook and other pages to get the information about new dates. Thank you very much for joining us tonight. I hope and I believe that you've gotten a lot of useful information. And I wish you a pleasant evening and a successful season in beekeeping.
Thank you. Bye.
First.
Thank you again.
Thank you.
Have a
Bye.
nice
Thank
evening.
you. Bye.
Goodbye. Bye.
Bye-bye.